All right, ladies and gents, welcome back. So, we've been having this discussion about the fact that your cells need to get materials, nutrients, oxygen, that sort of stuff out of their environment into the cell so that they can use it to be able to function. And then they also need to get rid of those waste products that are a byproduct of their metabolism. And all of this stuff has to happen across that cell membrane. So, when we look at how materials get in and out, there really are two major types of transport. And it really comes down to whether or not I need to use energy as a cell to get materials across that membrane. So, if we're looking at any type of transport that does not require energy, then we're talking passive transport. It's just going to kind of happen because the cell happens to be there. The materials, whether it's sugar or oxygen, just happens to exist inside and outside of the cells. So, what's going to end up happening in passive transport? All right, so passive transport here, no energy is going to be required, and I'm going to move materials from where there's really high concentration. And what that means is that there's a lot of that sugar in one place or a lot of oxygen in one place. And we're going to move it from where it's really high, where there's lots of it, to an area where there's not so much of it, where it's low. And so we talk about moving materials with their concentration gradient or along their concentration gradient or down their concentration gradient. You can kind of come across any one of those phrases, and it all means that I'm taking materials where there's lots of it and I'm moving it and spreading it out to places where there's not so much. And we're going to do that by moving those molecules from that high concentration to low, letting them spread out until they reach what we talk about as a state of equilibrium, meaning that they're equal on both sides. So I've got the same number of, say, oxygen molecules on the outside of my cell that I have compared to the inside of my cell. Now, the opposite to passive transport is active transport. Now I actually need to put in energy in order to make those molecules go where I want them to. And the reason I need to put the energy in is because now rather than moving them and just kind of letting them spread out, I have to actually make them more concentrated. I have to bunch them all together. And so we talk about moving materials either against their concentration gradient or up their concentration gradient. Those are some words we can think of. And so what I'm doing now instead is preventing those molecules from spreading out, preventing them from being equal on either side. So think of it this way. If I were to take a bucket of tennis balls and drop them in the middle of the classroom, right? Inside the bucket itself, lots and lots of tennis balls. When I dump them, I'm moving them from where they're really concentrated, right, in the bucket itself, where there's lots of balls, and they're being moved to an area where they're lower in concentration in the classroom on the floor. There weren't any balls there to begin with. So, what's going to happen? Those balls are going to kind of bounce off of each other, spread out until eventually they get pretty equally far apart from each other. That's when they've reached that sort of quintessential state of equilibrium. I don't need really any energy to make that happen. The balls are just going to fall, they're going to bounce off of each other until they don't hit anything anymore. That's the passive side of things. Active, on the other hand, is taking those tennis balls that are now all spread out and putting them back in the bucket. That means I'm going to have to get up out of my chair. I'm going to have to expend energy to put them from where they're already sort of low and spread out and at equilibrium and bring them back into the bucket where they're really tightly bunched together, putting them from high concentration, or excuse me, from low concentration where they're all spread out to a higher concentration in a smaller space. That's the big difference here. So passive, no energy. I'm going to take things where they're all bunched up and let them spread out, let them reach that equilibrium. Active, I'm going to need energy because I'm going to take them from where they're already spread out and force them to be bunched up into that higher concentration. As a result, preventing them from reaching that state of equilibrium. So today, let's talk specifically about passive transport, right? So let's focus there for today. And here we're going to, again, move those molecules from where they're really high to where they're really low. So if I look, here's my cell membrane. All right, I've got all of these, let's just say they're water molecules, lots of them outside my cell membrane, not really a whole lot inside. And so they're going to move down or move along or move with this concentration gradient, where they're really high to where they're really low. All right, and we're going to allow them to keep moving. And so they'll want to keep moving along this arrow until I balance them out. So if I happen to count, let's see, there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, and 15. So there's 15 water molecules, right? So what I really need is approximately half on either side. So 7 and 7 would get me to 14, and so we're pretty close. That's when we're talking about that state of equilibrium, where I've got the same number of one thing, water or sugar or sodium, on one side of the membrane that's equal to the other side. So when we look at passive transport in terms of how I can move things, actual processes I can use that aren't going to require energy, I really have three types, and that's this idea of general diffusion, osmosis, and then facilitated diffusion. So let's kind of walk through each of these. So if I talk about diffusion in general, all right, it really is just the definition of passive transport. I'm going to take molecules away from an area where they're highly concentrated, really packed together, and move them to an area where they're much lower, okay, less concentrated, more spread out. So when we define what concentration means, we're talking about the number of molecules of a particular substance, sugar, for example, in a given volume. So think of it like this. We talk about things like those Mio flavor packs, right? Right, you flavor your water with them. So if you look at it in the actual packaging that you buy at the grocery store, it's very concentrated. It's pretty much just all flavor and nothing really to make a true solution out of it, right? So it's mostly just the solute. It's all sugar and flavor. Now, to drink that would be kind of really intense. So what do? You put it into a bottle of water and you can spread out those flavor and sugar molecules and decrease the concentration, right? You're allowing that to diffuse. Another one you might want to think about is something like food coloring. You're dyeing Easter eggs. The actual dropper of food coloring itself is pretty highly concentrated dye. You stick it into that cup of water, and those dye molecules are going to diffuse through the water until it's all spread out and looks like a uniform color. That is diffusion. You've also experienced it in the locker rooms. Somebody goes and sprays a bottle of perfume or cologne or that Axe body spray, and it is initially concentrated, lots and lots of scent molecules right next to the bottle when it gets sprayed. Eventually, everybody in the locker room can smell the perfume because it's diffusing through the area. Those molecules are hitting each other, bouncing off of each other, pushing each other away until it spreads out across the entire locker room, and now everybody can smell it. That's diffusion. And by true definition, diffusion doesn't actually involve a membrane, right? It can happen in the air. It can happen in a cup of water with your dye for your Easter eggs. So the membrane technically isn't required for diffusion. Now, when we look at this in cells, the membrane happens to be present, and so diffusion of things like oxygen and carbon dioxide can happen across that membrane in the same manner. All right, and remember things like carbon dioxide and oxygen are nonpolar molecules, so they have no issue, I should say, interacting with those fatty acid tails of those phospholipids, so these guys diffuse straight across the membrane as if no issue. And that diffusion will continue to happen until we reach that state of equilibrium where everything was equal on both sides. Now, what if I need to move something that's polar instead, right? Nonpolar substances were cool. They're hydrophobic, just like these tails, so stuff goes straight through. But if I'm looking at something that's polar, so water or sugar, for example, your cells still need that material in and out of their cells. They have to be able to move that material with that concentration gradient, moving it from where it's really highly packed together to where it's low. But I can't move it through those fatty acid tails. The chemistry doesn't allow me to do that, right? Those lipids just repel the water back into the area that it came from. So anything that's hydrophilic, anything that's polar, what can I do about it? Well, now I can work with these transport proteins. So remember that these proteins allow me to have a hydrophilic environment in here so that they can interact with anything polar. So rather than going straight through the membrane like anything oxygen, carbon dioxide, anything nonpolar could do through simple diffusion, which is what we just talked about, now I have to have a protein in order to move that material, which means facilitated diffusion is a very specific biological process where something like this diffusion can happen with perfume or dye, okay, because I don't need a membrane. Facilitated diffusion only functions with a membrane in place, so it's a very specific biological process as opposed to just something physical that can happen anywhere in the world. So carrier proteins, transport proteins, channel proteins, whatever term you may come across, I need these guys now to allow me to move polar molecules across the membrane because they don't go through those fatty acid tails. So we can talk about a very specific type of passive transport in terms of osmosis. And osmosis is the diffusion simply of water across a membrane. Now, here's the interesting thing. We're going to define the solutions, the concentrations on either side of that membrane, really based on the solute, so sugar, salt, something like that. But when we look at the actual definition of passive transport, right, it's moving materials from where they're high to where they're low. And so we're going to have to take into consideration two pieces of information when we want to talk about where materials are going to flow when it comes to osmosis, because what often ends up being the case is that the solution is solute plus solvent, right? So water is our universal solvent. So in your cells, both inside and outside, there's lots and lots of water, and then dissolved in that is the solute, which could be things like sugar or salt. And a lot of times those solute molecules are kind of big and clunky, and so they don't really easily pass through the membrane themselves. So in order for your cells to get to a state of equilibrium, they can't move those sugar molecules, for example, across the membrane until they'll move the water instead. So the water is still going to travel from where there's lots and lots of water to where there's not so much water. But unfortunately, for whatever reason, science has decided that we're going to talk and define certain terms for how we discuss osmosis based on the solute concentration instead. So when you're looking at that, we're really moving water from where there's less solute to where there's high solute. But in order to achieve that, it's always 100% solution. So that means if there's less sugar, that means there has to be more water. Hopefully that makes sense. And the reverse would also have to be true. So if I take a look here in this example, right, I've got one sugar molecule on this side, three on this side. So because there's only one sugar molecule here, let's just say that accounts for 10% of the solution. That means 90% of it to get me to that 100% is actually water. So 10% sugar, 90% water. On this side, however, three solute molecules, so let's say that accounts for 30% of the solution, which means now I'm talking 60% water. So 90% water to 60% water, the water is going to move from here to here, but we define the solutions based on how much sugar or how much solute is in them. So that's where those terms are going to come from that we're going to see in just a moment. So water balance is really, really important for your cells. All right, in order for them to be able to move materials in and out and maintain homeostasis, they have to be able to regulate the amount of water outside compared to inside. But in order to regulate that water, it also means that they're having to take into consideration salts and sugars and things like that that also are existing in solution in their environment as well as in the cytoplasm inside the cells. So when we talk about the solutions or the environments that these cells can potentially find them in, we really end up with three major options. One we can talk about is an isotonic solution, and that prefix iso means same. So the concentration of water and sugar and salt is the same inside my cells as it is outside. We can talk about hypotonic, and that's this situation where now I've got not a whole lot of water inside, but lots and lots of water outside. And so that water ends up rushing in as a result because there's so much concentrated sugar or salt inside my cells that the water's going to rush in to compensate for it. How do you remember what's going to happen to the shape of the cell? You got this big O here, and that's going to kind of correspond to a big bloated, swelled up cell. So hopefully that kind of helps a little. And then you've got a third option where now rather than the water moving in or moving in and out at the same rate, I've got a hypertonic situation. So hyper means less. And so now there's less water outside compared to inside my cells. And so now that water is going to be moving out, sort of like packing meat in salt, right? So lots and lots of salt outside, not a whole lot of water. And so that water gets pulled out of the meat and actually preserves it, right? We shrink it, we shrivel it. And that's what's happening in that situation. So in an isotonic situation, right, I've got the same amount of water inside as I do outside, which also means I've got the same amount of sugars and salts inside that I do out. And as a result, my water can move in and out of my cells at the same rate. All right, they're happening to move 10 molecules of water in at the same time I'm moving 10 molecules out. And so your cells stay the same shape because they don't notice a difference, right? It's like you putting in 100 calories into your body that day, but you burned it off at the same time. So your body doesn't notice a weight difference. Consider this question. We take in water for hydration, right? To keep our cells plump and functioning. We need that water to survive. So why can't we drink salt water as a way to hydrate ourselves? We're stranded in the ocean, we're surrounded by water, but it's salt water. Why can't I drink that and help keep myself alive that way?
How big is this ocean?
Water, water, everywhere. Nor any drop to drink. Well, except maybe that drop. That's a little
Salty.
So as you noticed, the more we drink that water, all right, the more it's actually going to increase the salt in your surrounding of your cells, pulling the water back out of them. So rather than hydrating you and pushing water in, we're actually sucking the water back out. And so you can be surrounded by water in the middle of the ocean. And unfortunately, you can end up dying from dehydration. And that's because your cells are really ending up in this hypertonic state. On the flip side, we can talk about a hypertonic environment where the cells actually end up shrinking. They get shriveled up, and you may come across these terms. So, plasmolysis happens more specifically in plant cells, or we can talk about crenated cells, like your red blood cell here is a crenated or shriveled up cell. And what's happening is there's less water outside, more water inside. So way more sugar or salt on the outside compared to inside, and the water's rushing out. All right, the water's being sucked out. We're dehydrating it. We're shriveling it up. All right, and so the water is flowing out and causing the cell to shrink. Definitely not a good day for your cells. To be dehydrated is a bad thing. All right, you're not getting enough water to keep those cells nice and plump and functioning. And if they get dehydrated or shrivel up so much, they will eventually die. All right. Hypotonic is the opposite now. So much water on the outside, and so very little sugar that now all my water is rushing in to equalize out. And this can cause the cell, if too much water enters, to actually burst open, kind of like overfilling a water balloon. We talk about that term as a cytolysis. Cytocell, lyse means to break. So we're breaking the cell apart. All right, and this is because that water is just rushing in at crazy, crazy amounts of speed, and your cell has no way to stop it. All right, and as a result, it ends up busting open. So sometimes I find that it's really helpful for kids to see numbers to things. So let's just assume here's sort of my inside of my cell, my outside of my cell, and starch. These are big carbohydrate molecules, right? If I've got 50% starch on one side, 50% starch water on the inside, that makes my 100% solution. On the outside of my cells, however, I've got 70% starch. So that's where that hyper or that way more solute comes into play. But remember that I still need 100% solution. So 70% starch means 30% water. So what's that water going to do? It's going to go from where it's high at 50% to where it's low, and so this side of the water tube would go up, this side would go down. I'd be dehydrating my cells, they'd be shrinking. If I look at an isotonic solution, now 50% starch, 50% water still inside my cells, but in order to make it isotonic, I also need the same concentration, so a 50-50 on the outside. So water's still moving in both directions, it's just that we're not causing that water level to change. So your cells stay the same shape, everything's kind of good, off it goes. In a hypotonic situation, I'm going to still leave the same 50% starch, 50% water inside my cells, but now I need less solute, less starch. So something like a 25% starch solution means I've got 70%, 75% water. Well, now that water is going to move from where it's high to where it's low, so 75 to 50. Water level goes up on the inside of my cells, and the cell sort of swells up and gets bigger as it goes.